Okay, we are still working on this slide. We want to clarify some stuff and move on to even more new stuff. Um, so we are on the far left in photosystem 2. Remember, we are inside the thylakoids membrane. Light came in, so it's coming in. It is being captured by these pigments. So the green dots, the pigments, and they are the chlorophyll A, B, and the carotenoids that we're familiar with. And that then gets passed down until you get to this, it's a version of chlorophyll A, this P680. It just happens to be a very powerful oxidizing agent. And so with the input of water, that particular chlorophyll A, that particular one, has the ability to split water and give off oxygen, and then remove those hydrogen ions and put them into the membrane space. So into the stroma, so outside of the thylakoid membrane. And then that electron is excited and picked up by the primary acceptor. That primary acceptor, I think I misspoke on the last one. That is not NADPH yet. It's important that I make that clear. It is not. It is the primary acceptor. It then goes down this complex of proteins, and an incredible amount of ATP is produced. So the big product that's produced by photosystem 2 is ATP. Because we will need these to run the Calvin cycle. And then that same electron is picked up by the the next chlorophyll A in photosystem 1. That one also brings light in. That causes increase. It brings photons in, brings light in. That also excites that electron and is picked up by a primary acceptor, and it also moves down an electron transport chain with the help of this iron type product, which just went out of my head, ferredoxin. It's a protein. It's not an iron. It's a protein called ferredoxin. The reason I said iron is because fer is kind of the word we associate with iron, but no, it's a protein, ferredoxin FD. This particular chain doesn't create ATP, but this chain creates the NADPH, which is why it was very important I clarify this. This photosystem 1, the big product that it's responsible for producing is not only these hydrogen ions, but hydrogen ions that then can be attached to NADP in its excited state, and then you have NADPH. That can then go into the Calvin cycle. So out of these two, out of these two photosystems that you have in the complex that we were referring to. We just went through these photosystems are in the light harvesting complex. In the middle of the light harvesting complex, you have your reaction center complex. That's where the primary acceptor is. The two things that are produced are ATP out of photosystem 1 and NADPH out of photosystem 2. Those are both very, very important in terms of raw products that go into the Calvin cycle. The Calvin cycle is the light independent portion of the reaction that then produces the sugar. P680 in the excited state or the strong state is a very strong oxidating agent. Your book talks about it having a hole. When it has that positive on it, it has a big hole that is looking to, it has the ability to, I mean, enzymes split water molecules, and those electrons then are transferred from the hydrogen atoms to this P680 in its excited state, and thus reducing it to P680 not in its excited state. Sorry. Oxygen is released. The electron falls down the electron transport chain from primary acceptor of P2 to P1. Energy is released by the fall that drives the creation of a proton gradient across the thylakoid membrane. Proton gradient. So what we think of as higher concentration of hydrogens, they are going to move to an area of lower concentration. So that's how we're driving this concept of creating a proton gradient that is going to move those protons across the thylakoid membrane just based on a concentration gradient. That diffusion then causes ATP synthesis. Very similar to cellular respiration, that diffusion causes that turn of the cycle that allows for a phosphorus to be put onto ADP. So that causes that the photophosphorylation, which is a form of chemoosmosis. And for some reason I thought that was so important I had to say that twice. I apologize for that. That's actually twice on the slide. So that is how ATP is created. And then in PS1 the energy is transferred light energy, which loses an electron to the electron acceptor. So in PS1 it's P700, and again this is a very, very strong oxidizing agent, not quite as strong as P680 plus. That accepts that electron passed down via the electron transport chain. That electron, as it falls down the transport chain, from the primary acceptor of PS1 to that protein, and there you have the protein here written out, ferredoxin, that then is going to transfer to your electron acceptor, so that NADP positive, and then reduce it to NADPH. This is the very, you can think of it as kind of similar in the type of steps that it's done. So you have an electron come in, picked up by the electron acceptor, causes that that electron to become excited and picked up by the primary electron acceptor, and then down the electron transport chain via this protein, protein which is ferredoxin, and then eventually, as the energy becomes less volatile or less hot potato-ish, is going to be transferred to NADP. I left out a big piece, which you're probably sitting there scratching your head. They actually discovered let me go back, sorry. They actually discovered the photosystem 1 first. They knew nothing about photosystem 2. So they discovered photosystem 1 and assumed, as I said, they know most of what goes on in photosynthesis, but they discovered photosystem 1 first. That's why it's named photosystem 1, but it actually takes place in the second step. And then they realized that that it was actually oxygen that's being given off, and that oxygen is not coming from carbon dioxide, but is coming from the water. And that's when photosystem 2 was figured out or discovered, however you want to put it. So they happen in the order of photosystem 2 to 1, but they were discovered in the order of photosystem 1 to 2. That explains that mystery. And one of the reasons why I went back to that now, because I knew I didn't say that, is this cyclic flow only uses photosystem 1. So that was the first one discovered, but the second one that we talked about. And it produces ATP, but not NADPH. So with this system, you have ATP also produced, not as efficiently, but with just this one system, you have everything that then can go into the Calvin cycle. So this cyclic electron flow, and by the way, the electron flow that we just talked about is called the linear electron flow. This is the cyclic electron flow. Generates a surplus of ATP, and that then can satisfy the Calvin cycle's need for energy. But the thing about the cyclic electron flow is thought to have evolved before the linear electron flow, and so it's thought to be even older, and this might be a defense mechanism. It might protect the cells from light-induced damage. So what you have is the light coming in, being captured, sent to the primary acceptor. You have FD sending it through the complex and creating ATP in a cyclic fashion. And then after the complex then creates ATP, you still have a byproduct that can go back into the flow of the cyclic flow. Looks like I need to move my slide over a bit, but I can perhaps talk a little bit. I won't get all the way through this one, and so I'll fix it after I finish this teaching time. ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place. And so here you have inside the thylakoid space, you have this high concentration of hydrogen ions. That then creates this proton gradient that actually is going to work with ATP synthase to produce ATP from ADP. That's actually happening in the stroma because those hydrogen ions are getting moved out. That then is going to go into the Calvin cycle. With regard to NADP+, you also have that FD ferredoxin protein that actually is taking the NADP with the help of an enzyme, reductase, and actually attaching that hydrogen ion to it, and that then also can go into the Calvin cycle. These reactions are both taking place because of that photon gradient that was created by the light dependent cycle, but these are all happening in the light independent cycle. So in summary, the light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. And again, I apologize for this slide. The difference between chloroplasts and mitochondria, both of them generate ATP by a process called chemiosmosis. There you see the word right there, but use different sources for their energy. So they both do chemiosmosis to produce ATP. What do they mean by chemiosmosis? Is it's a type of phosphorylation where you are actually adding that phosphorus. You actually see the image here where you have this this proton gradient moving ATP synthase and attaching a phosphorus to ADP. We'll pick this up on the next one.